If you've eaten vegan burgers that taste like meat or used synthetic collagen in your beauty routine, both products that are grown in the lab, then you've benefited from synthetic biology. It's a field rife with potential, as it allows scientists to design biological systems to specification, such as engineering a microbe to produce a cancer-fighting agent. Yet conventional methods of bioengineering are slow and laborious, with trial and error being the main approach. Now scientists at the Department of Energy's Lawrence Berkeley National Laboratory, Berkeley Lab, have developed a new tool that adapts machine learning algorithms to the needs of synthetic biology to guide development systematically. The innovation means scientists will not have to spend years developing a meticulous understanding of each part of a cell and what it does in order to manipulate it. Instead, with a limited set of training data, the algorithms are able to predict how changes in a cell's DNA or biochemistry will affect. The possibilities are revolutionary, said Hector Garcia Martin, a researcher in Berkeley Lab's Biological Systems and Engineering (BSE) division who led the research. Right now, bioengineering is a very slow process. It took 150 person-years to create the anti-malarial drug, artemisinin. If you're able to create new cells to specification in a couple weeks or months instead of years, you could really revolutionize what you can do with bioengineering. Working with BSE data scientist Tatjana Radivojevich and an international group of researchers, the team developed and demonstrated a machine learning allows computers to make predictions after learning from substantial amounts of available training data. In ART, a machine learning automated recommendation tool for synthetic biology, led by Radivojevich, the researchers presented the algorithm, which is tailored to the particularities of the synthetic biology field, small training data sets, the need to quantify uncertainty, and recursive cycles. The tool's capabilities were demonstrated with simulated and historical data from previous metabolic engineering projects, such as improving the production of renewable biofuels. In combining mechanistic and machine learning models for predictive engineering and optimization of tryptophan metabolism, the team used ART to guide the metabolic engineering process to increase the production of tryptophan, an amino acid with various uses, by a species of yeast called saccharose. The project was led by Ji Zhang and Søren Petersen of the Novo Nordisk Foundation Center for Biosustainability at the Technical University of Denmark, in collaboration with scientists at Berkeley Lab and Teselagen, a San Francisco-based startup company. To conduct the experiment, they selected 5 genes, each controlled by different gene promoters and other mechanisms within the cell and representing, in total, nearly 8,000 potential combinations of biological pathways. The researchers in Denmark then obtained experimental data on 250 of those pathways, representing just 3% of all possible combinations, and that data were used to train the algorithm. In other words, ART learned what output, amino acid production, is associated with what input, gene expression. Then, using statistical inference, the tool was able to extrapolate how each of the remaining 7,000 plus combinations would affect tryptophan production. The design it ultimately recommended increased tryptophan production by 106% over the state-of-the-art reference strain and by 17% over the best designs used for training the model. This is a clear demonstration that bioengineering led by machine learning is feasible, and disruptive if scalable. We did it for 5 genes, but we believe it could be done for the full genome, said Garcia Martin, who is a member of the Agile Biofoundry and also the director of the Quantitative Metabolic Modeling Team at the Joint Bioenergy Institute (JBEI), a DOE Bioenergy Research Center. Both supported a This is just the beginning. With this, we've shown that there's an alternative way of doing metabolic engineering. Algorithms can automatically perform the routine parts of research while you devote your time to the more creative parts of the scientific endeavor, deciding on the important questions, designing the experiments, and consolidating the obtained knowledge. More data needed, the researchers say they. Yet to truly realize synthetic biology's potential, they say the algorithms will need to be trained with much more data. Garcia Martin describes synthetic biology as being only in its infancy, the equivalent of where the Industrial Revolution was in the 1790s. It's only by investing in automation and high-throughput technologies that you'll be able to leverage the data needed to really revolutionize bioengineering, he said. Radivojevich added, "We provided the methodology and a demonstration on a small dataset. Potential applications might be revolutionary given access to large amounts of data. The unique capabilities of national labs besides the dearth of experimental data," Garcia Martin says the other limitation Given the explosion of data in our world today, many fields and companies are competing for a limited number of experts in machine learning and artificial intelligence. Garcia Martin notes that knowledge of biology is not an absolute prerequisite, if surrounded by the team environment provided by the national labs. Radivojevich, for example, has a doctorate in applied mathematics and no background in biology. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.